All right. Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryoelectron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. I'm joined today by my colleague here at NCCAT, Ed Eng, as well as Sean Mulligan and Lauren Hales-Beck from the Pacific Northwest CryoEM Center, PNCC, and Lydia Joubert and Michael Schmidt from the Stanford SLAC CryoEM Center, S2C2. S2C2 has invited today's speaker, Christopher Koo. Lydia will introduce Christopher properly after my colleagues from each of the centers give a quick overview and update on the CryoEM resources that we offer at no cost to the research community. For anyone who hasn't attended our webinar series before, CryoEM Current Practices is an ongoing event that we host the last Thursday of every month at the same time. And we highlight the methods that researchers are using to obtain and interpret the data that they can collect at the national centers. So this month actually marks the 1-year anniversary of our series. And so I was looking back at our YouTube page that you can actually navigate to quickly using the tiny URL CryoEM talks. And you can see a great year of talks that we've had that dug into the power of CryoEM to answer biological questions. But we also really got into the weeds of what it takes to get those results. And when we kicked off this series, we had no idea how many people would show up. We thought, oh, maybe 20, 30 people will come, but we've actually had a much larger audience. And we're really excited to see the enthusiasm from the community. And we hope you continue to find this venue helpful for your own research and training. So back to the topic at hand today. We are recording today's talk, and you'll find that recording on that same YouTube page. And you can also find those recordings on cryoemcenters.org, where you'll also find registration links for future talks and general information about the larger NIH Common Fund CryoEM program. A quick plug for next month's talk. NCCAT will be hosting Helen Donalick from the Bass and Shen labs at the University of Utah. And she'll be talking about structural determination of the dicer 2 R2D2 complex. So we'll take a step away from memory proteins next month. So save the date for that if you'd like to join us again next month. A couple of final logistics. Please use the Q&A feature to send in your questions today. And if you have any questions directed to logistics or access at the centers, our panelists here will answer those directly in that Q&A box. And we'll save questions for Christopher to the end of his talk. And so with that, I'll turn over for quick intros from our center representatives. Ed Eng, you get to go first.
Okay, thank you, Christina. Welcome to NCCAT's new realm of access, in the sense that we are allowing users on site, and we have had many users access not only instrumentation access, but cross-training here. Currently, we have a couple of trainees right now. Something that's happening that we hope that will be very impactful is that come spring, we're going to hold our 2nd annual single particle short course. If you want to find out more information, you can go online. And lastly, what's really exciting, NIH has opened up a screening program. So we have a new grid preparation screening program that is going to go online right now. And if you'd like to find out more, November 18th, we have an AMA session. So go to nysbc.org, nCCAT.nysbc.org to find out more.
Thank you, Ed. And Sean, I'll turn it over to you next.
Hello, my name is Sean Mulligan, and I'm a microscopist at the Pacific Northwest CryoEM Center. At PNCC, we have 5 cryo-TEM microscopes available to researchers. Our 4 Krioses are equipped with the latest generation direct electron detectors. In addition to all 4 having K3 and Falcon direct electron detectors, 3 of them are configured with Catan energy filters, including the new BioContinuum. We offer comprehensive screening services that utilize our Arctica that is also configured with a Catan K3. For sample preparation, we have a VitraBot like a GB2 and are currently testing one of the latest generation vitrification robots. the cryosolve Vitrajet. PNCC offers one proposal type for single particle analysis and tomography that awards up to 480 hours a year. The hours are valid for up to 2 years. At PNCC, we have a monthly submission deadline for applications. Each approved proposal will be delegated a Pmoc scientific point of contact, such as myself, to assist you with your project. PNCC is currently under COVID restrictions, and we cannot accept on-site visitors at this time. In the meantime, we're offering small remote workshops, one-on-one remote training. Once we can have visitors on site again, we plan to offer on-site workshops covering microscope operation and sample preparation. Please visit our website for additional information and to apply. Thank you.
Thank you, Sean. And Lydia, I'll turn over to you.
Thank you, Christina. I'm Lydia Joubert, a CryoEM specialist at the Stanford SLAC CryoEM Center. S2C2 is co-located with the Stanford Synchrotron Radiation Light Source, SSRL, and the Line Coherent Light Source, LCLS, at the SLAC National Accelerator Laboratory at Stanford University. We occupy space in the new Arriaga Science Center, so-called Building 57. We recently installed 3 new Titan Cryos. We also have a relative humidity controlled lab with all the needed equipment for specimen preparation and evaluation, clipping and loading. In addition to user support in data collection, we also offer a rigorous training program that accommodates various levels of trainees, from inexperienced researchers who need support in sample optimization in an exploratory training program to customized personal training to suit the researcher's individual or institutional needs. We have 5 full-time CryoEM specialists, as well as IT experts and administrative staff to support all steps from shipping doers through booking instrumentation, advising users, clipping and loading samples, performing fully supported data collection, advice on data processing, and of course, tending to the smooth operation of instrumentation, which is often challenging. Of course, I didn't mention our leadership consists of Wachu, Mike Schmidt, and Brett Hedman. And then we host regular workshops on all aspects of CryoEM, from sample preparation through data processing and modeling. And you can find everything on submitting a proposal, registering for conferences and workshops, and also for our training program on our website. And then over to our speaker today. I actually met him during a Zoom session supporting his data collection a while back. Good to see you again, Christopher. He's a graduate student in Amy Rosenzweig's lab at Northwestern, and he's going to talk with us about methane-oxidizing enzyme in the lipid bilayer. And I'm not going to take any more of your time, Christopher. Over to you again. Looking forward. Thank you.
Hey, good to see you too, Lydia. Thanks so much for the introduction. And yeah, thanks so much to all the centers for putting on this webinar. I've learned a lot by looking at the past one. I hope that this can help contribute to the body of knowledge that's being gathered. So I've tweaked the title of my talk a little bit to get a little bit more specific about some of the things I'm going to be talking about, to handling and characterizing membrane metalloenzymes from native organisms. So as Lydia mentioned, my name's Chris, and I'm from the Rosenzweig lab at Northwestern University. So in the Rosenzweig lab, we're interested in bacteria called methanotrophs. Methanotrophs are characterized by their ability to use methane as their sole source of carbon and energy. And they have a lot of really interesting features, one of which is their sensitivity to environmental copper. And as you can see, in high copper conditions, methanotroph will generate these intercytoplasmic membrane structures that either stretch across the body of the cell or else along the periphery of the cell, depending on the species. And what's really striking about this is that it basically produces all this extra membrane layer similar to a chloroplast or mitochondria. And the reason for the production of this structure is this enzyme ticulate methane monooxygenase shown on the right. So PMMO is a copper-dependent metalloenzyme that converts methane to methanol and is essential for the life cycle of these microorganisms. So under high copper conditions, the methanotroph will produce those intercytoplasmic membrane structures and a very high amount of PMMO. Some species have the ability to switch to this soluble methane monooxygenase, or SMMO. Now, being a soluble, and this enzyme is iron dependent. And we'll basically wait until there's copper available in the environment to then switch to PMMO production. Now, there's a lot that's understood about SMMO because it's a soluble enzyme. It's sample handling, and there's been a lot of ability to dive really deep into the mechanism and structure of this molecule. However, there's been some roadblocks for understanding PMMO because it is a membrane protein. And one other thing is that we isolate PMMO from the native organism, so we are a little bit limited in our ability to do things like mutagenesis of essential residues in the enzyme. So to tackle this problem, we actually look at PMMO from multiple species of methanotrophs, and here's the metabolic pathway for methane. And down here you can see all these different species of methanotrophs grouped into 3 groups by the step at which they assimilate carbon into their biomass. But what all these groups have, they have this first step of methane to methanol conversion using one of the methane monooxygenases. And so by studying PMMO from different organisms, we can start to understand which residues are important and what their roles might be in methane oxidation and copper in PMMO. So these are some of the species I'm going to be focusing on the talk, mostly this Metallococcus species up here. And so getting to the purification scheme for PMMO, essentially what we do is we grow methanotrophs in a fermenter where we continuously feed methane oxygen, harvest the cells, lyse by sonication, and then isolate the membranes using different forces of centrifugation, and then isolate the PMMO from those membranes using detergent. Like I said before, PMMO is a really large component of these membranes, where once we isolate the membranes, it's 85 to 90% of all the protein that exists within those membranes. And this also means that we're able to get quite a bit of PMMO because it's naturally produced at such high levels within the methanotroph. Shown here on the right is, so what we've learned about PMMOs come from advanced spectroscopy of the metal centers, as well as crystal structures from multiple different species. So what the crystal structures have revealed is that PMMO is made up of 3 subunits, and 3 of those subunits form a single protomer, which is the catalytic unit of PMMO. And 3 of those protomers related by C3 symmetry form one PMMO complex. So looking at one of those protomers a little bit more closely, what we can see is the 3 subunits of PMMO. So PMMO A and PMMO C are almost entirely transmembrane in nature and are entirely embedded within the membrane bilayer, whereas PMMO B has this large soluble domain, as well as 2 transmembrane helices. Just a side note, I'm not going to go into too much detail in this talk, but there is this mystery helix, we're calling mystery helix, that comes along with some types of PMMOs. So this is one thing that differentiates some of the groups of methanotrophs, is that some of the PMMOs have this helix, about 20 amino acids, that crystallizes along with the larger complex. We haven't yet been able to identify the helix de novo. There aren't necessarily large bulky side chains where we can identify what the sequence is, and it's such a short sequence, about 20 amino acids, necessarily located within the genome, but we're really interested in trying to understand what this helix is and how it may be affecting activity in these specific group of methanotrophs. But nevertheless, kind of focusing on the hotspots of PMMO, there are 2 metal binding sites within one protomer. One of these sites is located in the soluble domain of PMMO-B, kind of sitting just on top of the lipid bilayer. And this site is coordinated by 3 histidines, as well as the amino terminus of the polypeptide. So this amino terminus is actually generated after signal peptide cleavage. And this is a common motif for copper binding known as the histidine embrace. And so these 3 imidazole groups, along with the amino terminus, coordinate the copper in a planar fashion. And I should note that there's been some debate through the years since the original crystal structure about the nuclearity of this site. And there's been growing evidence that this site is a monocopper site, and this work kind of contributes to that, some evidence to that as well. Moving into the membrane bilayer, we also have the copper C site, which is located in the PMMO-C subunit. And this site is coordinated by 2 histidines as well as an aspartate. And comparing these 2 sites and trying to understand the role of the 2 sites, what is noted is that both of these sites are highly conserved across all species of methanotrophs. However, the copper C site is strictly conserved in every single PMMO that's been found, whereas the B site is conserved in both group 1 and group 2 methanotrophs, which have been really well characterized, but is not conserved in the group 3 methanotrophs, which is a newer group of methanotrophs that have not been as characterized as fully. So that leads us to believe that both sites are important for activity, but that this site is the site of methane oxidation within the bilayer. Concerningly, though, what's been missing from all the crystal structures, in every single crystal, in all 5 crystal structures we have from 5 different species, there is a stretch of amino acids that's missing that is right near the copper C site. And so this stretch is always at least 25 amino acids. And as you can see here, is really is right next to the copper C site. So it leads us to wonder if we're missing something, missing some information about the copper C site and about the active site of this enzyme. So it's really critical for us to understand the structure in this region to understand methane oxidation by copper. To drive that point home just a little bit further, this is a sequence logo of 300 inoperon Pmoc family subunits. What you can see are the strictly conserved residues are colored in purple. And the yellow bar here underlines the missing region. So within this missing region are some strictly conserved residues, as well as some metal binding residues. And I should note that this other region here of strictly conserved residues actually corresponds to the copper C site. So it's really critical that we understand what's going on in this region to understand methane oxidation by this enzyme. So just to summarize, these are kind of the outstanding questions that we have. So what is the structure of this highly conserved loop? And then how that leads to what is the biological active site structure of PMMO? Furthermore, being a membrane protein, most of this work has been done in detergent, and so I've been really curious what role the lipid bilayer may be playing, or specific lipids in that lipid bilayer. And then, as I mentioned before, just the nuclearity of those copper sites. So to tackle these questions, I started off by using Nanodiscs as my platform of choice for reconstituting PMMO into a lipid bilayer. So nanodiscs are a lipid bilayer that is bounded by a membrane scaffold protein, rendering the PMMO embedded in a lipid bilayer into a soluble particle. And this nanodisc is formed by a mixture of lipids, membrane scaffold protein, and PMMO, all solubilized in detergent. And upon detergent removal by biobeads, the nanodiscs self-assemble around a lipid bilayer containing PMMO. Now, a few things to note here. We found that it was essential to add copper during reconstitution to recover activity. And that's kind of interesting, where there is some copper that's lost through handling of PMMO, and so adding it back during reconstitution but not after was important for getting activity back. Furthermore, we also altered the lipid identity as well as the identity of the membrane scaffold protein. I'll get to that in the next few slides. So just a side note, so first talking about the which membrane scaffold protein we chose. So originally, we had some trouble embedding PMMO into these MSP1 E3D1 large nanodiscs. And so that led us to believe that although it looks like the disc should be big enough to fit PMMO, it led us to believe that perhaps it wasn't large enough. And so we actually started using these MSP2N2s, which are the larger, even larger discs than MSP1s. And I was able to get PMMO embedded into this disc. However, the activity assay that we used to determine methane to methanol conversion by PMMO occurs at 45 degrees for this species. And so using size exclusion chromatography, what I did was look at the stability of these nanodiscs. And as you can see, after just 5 minutes, the MSP2s were pretty much destabilized at that temperature, whereas the MSP1s were highly stable up to 12 hours. So this is just a note to say that the MSP2s, while they have a little bit of thermal instability, the MSP1s are also very, very stable if that's something they're looking for in your project. So really critically, upon copper addition into the nanodisc, we're able to see a significant increase in activity of the PMMO particle, significant recovery activity as compared to methanol production with PMMO in the detergent micelle. So here's where I'll talk about the different lipids that we used for reconstitution. We used both DMPC and POPC, which are common lipids used for nanodisc reconstitution. But we went a step further and actually isolated lipids from the methanotroph organism and then fed it back to the reconstitution. and then basically had nanodiscs that were in an even more native-like environment, and did see an increase in activity when we did that step. So we were curious as to why we see these variable levels of activity with different lipids. And so we did some mass spec analysis for the native lipids to look at, and we were able to identify the head groups that are present in this sample. As you can see, there's a variety of different head groups compared to the synthetic lipids. Additionally, this work that the native lipids have a mixture of both saturated and unsaturated acyl chains. So what I believe is happening is the POPC nanodiscs are a little bit looser packed, a little bit more closer to native than the DMPC nanodiscs, and therefore confer more activity than the DMPC discs. However, something in the native lipids is actually conferring even higher activity. It could be this cardiolipin, or else something that's missing. And so work is continuing to be done in our lab by Frank Tucci to understand more specifically what could be contributing to that higher activity. So of course, the next step was to look at, is there a structural reason for this recovering of activity? And so to do that, I turned to CryoEM of this sample. So this is just an example micrograph. I'm showing the particles in ice, as well as the 2D class averages that emerge from that data set, where you can see the soluble region of PMMO kind of emerging from the disk at multiple different angles. This is a gel showing 3 different subunits of PMMO, as well as the membrane scaffold protein polypeptide. And these are the parameters that I used to generate this data, where I used VitroBot with 2-sided blotting, 3 microliters at 1 mg per ml. And I was able to use open hole with C-flat 1.2, 1.3 spacing. So one benefit of this project is that we do get, by growing the methanotrophs ourselves and isolating PMMO, we do get quite a lot of sample, not necessarily sample limited. So here is the data processing workflow that I chose to use with Relion. So starting with about 9,000 movies, used a pretty inclusive picking regime to get about 4.5 million particles from there. And this step of 2D classification, and use steps of 2D and 3D classification to get rid of noise. And this is important because, again, using the native organism, PMMO doesn't have a tag, doesn't have an affinity tag, so we're not necessarily able to separate out sample-wise filled disks from empty disks, but computationally we're able to do that. And also we used a ratio of membrane scaffold protein to PMMO that was close to 1 in order to limit the number of empty disks that we obtained. So doing 2 rounds of 3D classification and picking the PMMO classes results in this many particles for the initial reconstruction. Here we can see the disk. And then just after a round of CTF refinement, particle polishing, another round of CTF refinement, I was able to get to 2.22 angstroms. And then employing basically tighter and tighter masking, sort of imitating the methodology from CryoSpark, we're basically able to get down to 2.15 angstroms for the structure. So looking closely at the map colored by a local resolution using Relion's implementation, you can see that there's not a lot of flexibility in this structure. And the flexible regions are really just at the periphery of the enzyme. This structure is just highly stable and just really well resolved throughout. So next I'm going to walk through the structure and look at how Cryo was able to tell us really interesting things about the different metal sites, and how it was really important to look at PMMO in its native environment and the active species of the enzyme to understand the architecture of the active site. So here's PMMO with the disk. Just removing the disk, we see the post-process map that's embedded within the disk. And then this is the resulting model from that map. So first off, looking at the copper B site, again, just reminding you that this site is located in the PMMO B subunit, sort of at the near the surface of the membrane bilayer. And what we can see is it's coordinated by these 3 histidines as well as the amino terminus. And we're actually able to resolve 2 waters in the active positions from the planar coordinated copper. Giving a little spin, you can see the waters here. And what's really important from this as well is we can see that it's clearly only 1 copper is able to be fit at this site. So this just provided more evidence that this site is monocopper. And if we then threshold the map up a bit, you can just see that the density here is basically very persistent, and then also resolves down to a single sphere. indicating that there's just one copper at this site. I should note that also that this site, the histidine at the amino terminus is a little bit less well ordered. This is consistent with what we see in crystal structures where because this is the N terminus of the polypeptide, this has a little bit of flexibility. It's not quite as well ordered as these, and not quite as persistent as these other residues. So zooming back out, what was really striking about this structure is that we're able to see stabilized phospholipids throughout the structure. And here what I'm showing are lipids on the periphery of the enzyme, where even in this zoomed out state, you can see two tails of lipids as well as the head group located sort of in the crevices between two protomers of PMMO, as well as these ones on the outside of one of the protomers. But even more strikingly, if I make the model transparent and now look on the interior of the enzyme, we're able to see for the first time this bilayer within the enzyme. So one thing I didn't mention is that these three protomers of PMMO form a pore in the middle. And for a long time, we didn't know what was happening in this pore, whether it was filled with lipids or not. And certainly there are other enzymes that have a pore, and there have been cryo work to show that there is a lipid bilayer in there. But for the first time, we're seeing this well-ordered lipid bilayer within the pore. And this lipid bilayer, specifically in the interior, has really big implications for the copper C site that's located within the membrane. So now what I'm going to do is just look at a single catalytic subunit, just look at a single protomer with the subunits in different colors. So the Pmoc in gold, Pmob in purple, and Pmoa in teal, as well as the interior bilayer. And I'm just going to look at a few well-stabilized lipids within this interior bilayer that are abutting this protomer. And if we zoom in now and we look at the copper C site, are these well-resolved lipid tails that are, again, part of the interior bilayer and are abutting the Pmoc subunit. And what's really critical for this cryostructure is if I now hide the A and B subunits and just look at the C subunit, what I've highlighted here is that highly conserved region that I was mentioning before. And we can see just how this highly conserved region is abutting the membrane layer and the role that the bilayer is playing in folding this region. So if we take another look at it, we can see how the bilayer basically helps fold up this region, and then also how the previously unmodeled residues form this 4-helix bundle at the copper, stabilized by the interior lipids. So going a step further, if we now look at the residues that are stabilized, what's striking is that we get a complete picture of the architecture of this copper C site for the first time. And what's even more striking is that if I zoom in, what I showed before, this on the right is actually the crystallographic site. And on the left is completely new residues coordinated to what looks like a metal density on the left here, which we're calling the copper D site, as well as waters that we can model in the structure as well. So looking at the map, you can see that what's really interesting about the copper C site is that there is no clear metal density located in the site, but there is a water density. The histidine that was previously coordinated to the copper is now bent out of position, out of metal coordinating position. Furthermore, there's a very strong density in this copper D site, as well as this water that comes along whenever the copper D is occupied. So thresholding it up again, you can see how this site reduces down to a single atom again, and how you can still see the clear positions of all these residues. So for the first time, this really gives us a more complete picture overall architecture of the active site. And it's really striking because it looks different than what not only does it look more complete than the crystal structure, but also introduces possibly a new metal binding site with those, again, strictly conserved residues. Just to give you another view of this, here's a comparison of the crystal structure to the CryoEM structure. And in the crystal structure, you can see there's just a dearth of information where all these residues were disordered and unmodeled. In the crystal structure, I should note, for all crystal structures other than this one, this is a copper. But this particular species has a zinc modeled due to a very high level of zinc in the crystallization buffer. And And you can see that this arginine is kind of pointed in the wrong direction, and there's just a lot of missing residues. And in the cryostructure now we can see all these residues as well as some hydrogen bonding networks that are actually positioning this arginine in the correct position. And just to give you just one more view of the resulting model, on the left here is just the kind of incomplete crystal structure model, and on the right is now the complete CryoEM model of that active site. And this is just providing more evidence for this being modeled as water, because it is within hydrogen bonding distance of a few residues. That's one more reason why we model as water at this site. It's also really interesting that we're able to see strong density for this water down here. It could indicate that this is a placeholder for something like methane, methanol, or even oxygen. Although we obviously can't make that determination just based on density alone. So this led us to the question, so now that we see this different architecture of the site, what is the biological occupancy of the site? And to answer that question, I looked at other structures from this species. So PMMO goes through a lot of different steps in terms of handling to go from isolating from the membrane and reforming to the disc. And copper loading can be challenging throughout that process and can result in samples that look biochemically identical in terms of activity, in terms of copper number, but then give us some slightly different results in the cryostructure. So this high resolution really made it possible to look at really minute differences in the active site of these different structures. So here's the 2.1 angstrom structure that I solved, where if we threshold up the map, you can see that now actually the copper C site is occupied, and the histidine is now in a coordinating position for this copper C. If you give it a little bit of movement, you can kind of get a better sense of the nuclearity of the site. But notably, what you can tell by thresholding up the map is now that loop is actually unstructured in this particular sample. And so it kind of leads between the crystal structure and this cryostructure, it kind of leads to this correlation of the loop region being disordered and then the copper C site being occupied. And so it's kind of interesting where it could suggest that this actually is an artifact, and that when this site is properly folded, this is the proper copper binding site, and this is meant to be empty. But it still raises the possibility that there could be copper at this site in the biological, in its native environment. One more structure actually suggested that we may have dicopper, and so there's a lot of caveats to this, obviously. But some of the reasons for this conclusion for this structure are that there's three reasons. One is that the water that was previously modeled in site is now missing. The histidine is now actually in a copper coordinating position, and if I spin the map, you can see that there is some density that's coming off of this histidine that could be a copper. So it just raises the possibility of a dicopper site in this site, in this active site. And so we're still looking for more ways to conclusively identify metals at these two sites, as well as understand what the biological occupancy of these sites is. So as I mentioned before, we do study other PMMOs from other species to answer some of these questions. And so I did study this other PMMO from this species 20Z. Didn't get to quite as high resolution, but this structure does illustrate the effect that the nanodisc is having, where the crystal structure solved in detergent had quite a lot of information missing from the PMOC subunit, whereas once embedded into the nanodiscs, I was able to model all of the missing alpha helices in the PMOC subunit. Looking closer at the copper C site, I was able to fit residues, and then also the copper C site looks occupied by copper at this point. But there is still some disorder, such as not being able to model the arginine side chain. And this site, this structure is not actually active. So it kind of, it raises the possibility of it contributes to the possibility of copper C site, but it's not necessarily the in vivo occupancy. Looking again at one other structure from species Rockwell, this species is active, and again, the loop region is not as well ordered in this structure, but it does have a density that looks like copper at the copper C site. I also note that in this case, deep enhancer was helpful to actually model these residues. So I'm not showing that map, but that map actually helped me really clearly model these residues as well. But it kind of indicates that there is some flexibility and disorder in this region, which could be leading to loading of the copper C site. To go into a few more things that I tried to process this data, commonly subtracting the disk out, although subtracting the disk did not lead to high resolution, and also looking at the copper C site looked pretty much identical when I subtracted the disk and solved that structure. Looking at different sets of frames of the movies, for example, omitting the first frame, was able to do that, but it also didn't look very different. And then one thing that I've tried a lot is symmetry expansion, where masking out a single protomer to try to look at protomer differences. And so far haven't had success with this because essentially I'm able to subtract out one protomer, but everything classifies into one class when I do 3D classification without alignment. And so to me that indicates that because there's not a lot of flexibility, and because that loop region is ordered and is not necessarily forming specific conformations, and it may not be large enough and it may not be distinct enough to separate out classes of protomers. But I think there's still more strategies in that direction to be applied. And also, I did this work in CryoSpark as well, and did the non-homogeneous reconstruction, and got a very similar looking molecule and very similar looking Pmoc site as well. So, one last thing I'll talk about that we tried was to look at the effect that copper was having by removing copper from the structure and then adding copper back to try to really look at the specific function of copper for the structure of this enzyme. And so there's been a well-established procedure in our lab where we treat the membrane with calcium cyanide to chelate out copper. And there still is some copper left in the sample. This is likely located in the PMMO site, which is which we've shown has a much higher affinity for copper. But upon treating with cyanide and then isolating PMMO and reconstituting to a disc, I was able to see that there was a severe reduction in copper for this sample. And then what I did in parallel was to treat with cyanide and then add copper back to the membrane, which we've shown restores catalytic activity. And then upon isolating that PMMO from the membrane and then reconstituting into a native nanodisc, I was able to get more copper in that sample. Structural analysis, so this was really striking to me. I was able to actually solve the structure of these two different samples, albeit at lower resolution. So the cyanide treated sample looks like this, and it actually is missing quite a lot of information in the Pmoc region. Really notably, the B and the A subunits, I was able to build a model for the entire A and B subunits, but the C subunit is really clear here is missing a lot of information. And that missing information begins and ends in two places at copper coordinating histidines. Strikingly, when I added copper back to the membranes and then solved that structure, I was then able to see that the alpha helices of the Pmoc subunit sort of reformed their structure, and I was able to model the entire Pmoc subunit. So this kind of shows the effect that copper is having in stabilizing the structure of the PMMO subunit, and specifically the Pmoc subunit. So at 3.5 angstroms, there's a lot of caveats as far as looking really closely at the at residue or metal information. But just looking at the copper C site for this structure here, if we look at both the C site and the D site, you can see that I'm able to model to fit residues into the map. And there is the suggestion that there could be a metal density coming off of this histidine and that the C site is empty. But this obviously has some caveats because it is at low resolution. And the takeaway really is that the copper is helping to reform that C site overall. So, so yeah, that's kind of the conclusion of the findings for this project. So this really, I think, illustrates the usefulness of CryoEM for membrane metalloenzymes, study enzymes in the native environment, and just shows how the nanodisc and the membrane environment had a really dramatic effect on this enzyme to the point that we were able to see this really, really critical region that we couldn't see before, and how CryoEM really enabled that by being able to study the enzyme in its native environment, whereas we couldn't really do that with crystallography. And so down here are kind of the two possibilities at this point of the native occupancy of that site, but we're looking for more ways to try to clearly identify what the native occupancy is. Now that we have the architecture, really understanding what the native occupancy for methane oxidation by copper. So yeah, we were able to restore catalytic activities in nanodiscs and see these highly conserved residues now. as well as determine that the B site was mononuclear and really able to see the lipid bilayer within the pore of the enzyme for the first time. So the further directions are really to conclusively identify this metal, and then look at just the native occupancy of the site, as well as look more closely at what the actual of the lipids that are bound. So with that, yeah, thanks very much for your attention. Thanks to everyone in my lab, especially Frank Tucci, who's worked really closely with me on this project, and the HE lab at Northwestern, which has given me a lot of, has really helped me get this off the ground. They have a lot of expertise in CryoEM and has helped me in roadblocks with the project, as well as I couldn't do any of this without the core facilities at Northwestern, as well as these people in particular that fall under various groups. And of course, thank you so much to the CryoEM centers who some of this data was collected at each of the centers, and none of this would have been possible without the really high quality data that was collected there. And thanks so much to the funding sources from the NIH and as well as the molecular biophysics training program at Northwestern. So yeah, thanks so much.
Awesome. Thank you, Christopher. That was really, really gorgeous. Those ordered lipid tails. I'm drooling over here. And I'm going to let Mike go ahead and moderate the Q&A. We've got a couple questions in the box already, but go ahead and send in your questions now.
Okay. Great job, Chris. Thank you for that great talk. We have a question. Could you explain more in detail how you perform the 3D classification to find the different classes of the copper C site? Do you provide, in general, when you're trying to do the 3D classification, do you provide a mask that just covers that part, or did you try 3D classification on the whole object?
So 3D classification was done using C1 symmetry. So it was all done in C1 and using the entire particle. So I didn't try anything beyond that to get that together class. And I wasn't sure if this is being asked, but those 3 C sites that I showed were actually from completely different samples of PMMO and from different data sets, essentially.
So they weren't 3D classifications of data set. They were different structures. Yeah.
Exactly. Yeah.
Mm-hmm. Very nice. This is an interesting question about the substrate possibilities. What could be added as a stable replacement for methane to see the substrate site? Is there any? Are there any kind of substrate analogs or something you could put in to see what's happening with the methane?
There are some substrate analogs that are also very small and are maybe difficult to see as well. One strategy that we're thinking about applying in the future that's been applied to the SMO I talked about is to use a longer chain alkane along with maybe some groups on the alkane that are a little bit more electron rich in order to identify a little better. So there's been work before to show that PMMO can oxidize maybe up to like hexane or heptane. So something with maybe a longer chain may be able to capture it in that site and sort of understand where the substrate's binding based on that. That's kind of the strategy. That's one of the directions that we're going.
Cool. What's the typical turnover rate in this particulate MMO?
So the turnover rate that I've seen is around 3 to 4 turnovers, and so we lose turnovers whenever we go from so obviously from whole cell to membrane. It's about, I think, a 10-fold loss in turnover, and another not quite 10-fold loss when we go into the nanodisc. But we are seeing multiple turnovers in the nanodisc, and it's especially dramatic compared to what we see in detergent micelles.
Mm-hmm. Okay, and a question about the copper levels. Number one, was the crystal structure done in presence of copper and about the same amount of copper as you had in the nanodiscs? That's one question I had.
Yeah. So like I showed with this particular species, zinc was found to be necessary for crystallization. So there were much higher levels of zinc actually as to what I'm adding to my nanodiscs. But there have been other species that have been able to crystallize in the presence of copper, as well as species that we crystallize without any metal at all and still see copper at that site. but it's definitely the levels that I'm adding to the nanodisc are much lower than the levels that are in the crystallization buffer.
Mhm. And in this critter, does it actually concentrate copper in the inside the cell, or is there a copper transporter? And what might be the level of the concentration of copper inside one of these methanotrophs?
Yeah, that's a good question about the actual level within the methanotroph. I don't have an exact answer for that specifically, but I will say that along with those intracytoplasmic membranes that are generated within the body of the cell, they also, under low copper conditions, have this entire machinery for basically exporting a really strong copper chelator called methanobactin. And this is a post-translationally modified peptide, which has a lot of enzymes that do all that PTM to make it really strong for copper binding, which it'll export into the environment to grab any copper that's there and then import it back into the cell. So it has a lot of very specific machinery to sustain such a high level of expression of PMMO and get as much copper as it possibly can to do that, to sustain this lifestyle, essentially.
Mhm. And I guess that means that the soluble MMO is not quite as good or not quite as advantageous as this particulate one anyway.
Right. I mean, part of it's also that, PMMO is embedded in the membrane, and there have been some studies to show that one reason why it's in the membrane is that methane tends to concentrate in the membrane. So that's one method of getting this really small substrate where it needs to go, is actually just going to where the substrate likes to concentrate. So that's one reason why the PMMO would be more efficient. Yeah.
Okay. We
Actually,
don't have.
I might pop in with a question if I can.
Go ahead. Yeah.
I had two questions actually. My first one, so you're going back to those ordered lipid tails. Do you think there's some sort of classification happening that's pulling out particularly ordered lipids, or are they really just, it's just like actually a very specific orientation of those lipids against your transmembrane helices that's very specific?
So there are some highly conserved residues on that PMMO and in that region that I was talking about that are actually some hydrophobic residues that are sticking out and contacting to those lipid tails. So there could be some really high order structure in that region that's causing those to be ordered. The ones in the crevices, I think the ones that are in the crevices on the periphery of the enzyme are really, really interesting because they do just sort of fit right in between two protomers. And I think they have something to do with conferring structural stability overall to the enzyme. But I don't know. I mean, there's definitely some lipids that look a little bit more smeared out towards the middle of maybe the bilayer, where it just kind of looks like a bilayer. So I don't know if as you get towards the center, if they're quite as well ordered or anything. But those ones that are right next to PMMO, I mean, there's still a little bit of noise associated with them, but it does look like some of them are like really, really highly stabilized by that section.
It almost looked like some of them were helping stabilize your copper D site.
Yeah,
Like the
exactly.
lipids themselves were sort of like pushing
Yeah,
those
exactly.
helices. Yeah.
I do think that's sort of what's happening. That's one thing that's happening is the lipid bilayers kind of exerting the correct forces on the enzyme to kind of fold up that site and complete the four helix bundle to complete the architecture.
And actually related, your alkylophyllum structure that was less ordered in that region, what
Yes.
kind of lipids were in those nanodiscs? Were you also using sort of a native lipid mix, or was that
So the native
different?
lipids we only used for the structure that I talked about the most, but for this one we only used the synthetic POPC lipids. So yeah, we may have more success if we were to isolate lipids from this species as well. So we may actually get activity from the bacteria as well.
Oh, thank you.
Any more questions from the attendees? Just put it in the Q&A.
And I've got another one while people are typing. Have you considered trying to solve a structure in detergent and see if it looks like the crystal structure? I could come up with a million reasons why not to waste your time doing that, but as a thought experiment.
Yeah,
Would that be worth doing?
I guess I was so focused on just getting PMMOs into discs that I didn't do that. Yeah, that I think we've kind of moved past that a little bit.
Yeah.
Yeah, but it would be interesting to see what it would look like with detergent. There actually has been one published in detergent, and from that structure, like it actually essentially looks like this, where most of this region is like completely is not there. The Pmoc site is not visible at all. So the most highly conserved part of the polypeptide is not conserved in that structure. So we kind of moved beyond that.
Oh, here's one more question. Oh, from Amy. Do you see the lipid bilayer, the cyanide treated sample structure inside that pore?
Right, that's an interesting question. So for the sample that was treated with cyanide, most of the lipids are absent. So that sort of indicates that there's some back and forth relationship between structure in this region as well as the lipid bilayer. So perhaps the ordering of this site by copper actually helps recruit lipids as well. So it's kind of this, maybe equilibrium is probably the wrong word, but both are necessary to fully reconstitute the native environment. Whereas when I have the cyanide treated one, I do see lipids on the periphery and one or two on the interior, but it's not as clear as that high resolution structure that I had.
Mm-hmm. And then a question: what is the role of copper in catalysis? Would iron work? That is,
right. So iron does work because iron works in the soluble methane monooxygenase, but iron doesn't work in this enzyme. I've also done this studies reconstituting with iron, and we've done lots of extensive studies in the membrane where we can just basically chelate and add iron back, and we've seen that doesn't recover activity. So that's why we think that those ions are all copper, and that is just strictly copper dependent.
Okay, and I don't know the acronym, but SMALPS might stabilize more than detergents.
Yes, yes, we are exploring that extensively as well. So yeah, the SMALPs are from someone that's handling nanodiscs quite a lot. The SMALPs are very nice because they do everything I'm trying to do in terms of reconstitute native lipids and all that quite nicely. We've hit a roadblock where we're actually not getting activity from them, so they might do too good of a job of isolating out of the membrane. But that work is still ongoing because the SMALPs are really fascinating. Would be a really good tool for this as well.
What does that acronym? What does that acronym mean anyway? I'm sorry.
Styrene acid lipid polymer lipopolymer. Yeah, I think that's what it is.
Okay, fine, maleic. Okay, maleic, small things. Okay. One more. Does the community know of any way to identify specific metal ions in density maps? Oh, that's an interesting one. we just had a validation challenge for trying to identify waters versus metal ions versus and mostly it was to distinguish waters from the metals. We didn't get down to parsing which metal was which. It's a little bit more. It's mostly would have to do with the coordination. This is from our challenge that we undertook with the PDB. It mostly has to do with the coordination geometry. If you recognize the coordination geometry of zinc, which is usually tetrahedral, versus copper, which I guess has several different kinds of coordination. It's mostly the coordination and the bond lengths. Metals just have a shorter bond length than hydrogen bonds to water. But anyone else in the community would like to chime in on how to distinguish like a copper from an iron from cobalt. Those are pretty difficult. Anyone?
I haven't heard of anything. So something for us to work on.
Yeah, I'm not sure it's even clear in X-ray crystallography how that exactly is done. Density maps have generally aren't really well distinguished by their density values as to what kind of molecular weight is involved. So it's a difficult problem. Yep, that's right. In X-ray, you use anomalous scattering. Amy chimed in. Yes, for some of the heavier ones, yeah, you can do that, and yeah, that also even it's possible. Okay.
So yeah, we're definitely open to any suggestions about using this data to try to identify before we move on to more advanced spectroscopic techniques to try to identify those metals. But anything, yeah, from this would be really really welcome.
All right. Well, if we have no more questions, Christopher, thank you so much for that fantastic talk. We will certainly be keeping our ears peaked for any way people might be coming up with ways to identify metals using cryo-EM data. But the difference in the way that electrons interact with proteins compared to X-rays is a bit of a challenge in that respect. And someone just posted one suggestion in the chat that we can look at. But with that, I will actually say thank you to Christopher. Thank you to everyone for joining us. We hope we will see many of you as users at any of the national centers. And we hope you join us again next month. And good luck with all of your cryo-EM endeavors. Thank you.
Thank you.
Thank you.
Thank you, Chris.